So let's whiz our mind back to 1970. Okay, Elton John's in the charts, topping everything. The Candle in the Wind has just come out. What we thought, how we used to classify these organisms, was we had sort of like uh, life. Life could be classified into these 5 kingdoms. So we had animals, we had fungi, we had our dustbin of life, the protists, we had the plants, and then we had these bacteria. So bacteria, which this is what's confusing, is you don't know about one type of these bacteria. So you've got your eubacteria. So these are this in real terms, these are your eubacteria. And you've got your, or as they were known at the time, these archaebacteria. So archaebacteria are not what we would sort of constantly refer to as your normal bacteria. They're weird and wonderful organisms that we don't learn much about at school, but they look like bacteria. They look like little rods if you look at them under a microscope, but they tended to be slightly different. They were in very extreme environments. They sort of lived in underwater vents in extreme heat. Underwater vents, or you found them around volcanoes. So there were these weird and wonderful organisms that looked like bacteria, but were found in different areas where you wouldn't expect eubacteria, our normal bacteria, to live. But these could both again be classified as prokaryotes because they both don't have a nucleus. So these are prokaryotes. All these other guys over here, the other 4 kingdoms, so bacteria being one kingdom, and these 4 other kingdoms have nuclei. So these are eukaryotes. And this was how we lived. This is what we knew. This was what the 1970s, what we thought in the 1970s. However, as we found out more about our favorite molecule, DNA, did DNA analysis. And what we found with this DNA analysis is that these archaebacteria had, so they didn't have a nucleus, but they still had DNA. If you stretched out their DNA, their chromosome, they would have genes dotted in between. So gene, gene, gene. And in between, they would have non-coding DNA. They'd have these stretches of A's, T's, and C's, A, T, C, G, etc., which didn't code for proteins, so they weren't genes. So the one example you might have heard of these are promoters. So this is non-coding DNA. The weird thing about this, and the reason this put a cat amongst the pigeons, changed our thinking, was that eubacteria, normal bacteria, bacteria that you learned about in 4th form, don't have this. Their DNA is circular, not straight like I'm showing it, but just for the sake of this video, I'm showing it like this. It's just end to end. It's just gene, gene, gene for the most part. Okay, so they have no non-coding DNA. And although some of you might not realize it, this is actually a massive difference. The whole way that their genetics works is completely different. And even more interestingly is that the archaebacteria, so the archaebacteria up here, this is very similar to the eukaryotes. So eukaryotes have so all the plant, fungi, animal, protists have DNA like this, whereas bacteria, so our eubacteria, sort of were on their own. So rather than have archaebacteria as a subcategory, almost like a cousin of eubacteria, like we did all those years ago, we rethought actually how things should be arranged. and we brought in these new names called domains. So now Carl Voser has done Carl Voser did all this research, or Vosser, I'm not entirely sure how to pronounce his name, and he sort of depicted it like this instead. So if we zoom in over here, we had life, and you had the prokaryotes, which were your bacteria, or in this case are eubacteria, with no non-coding DNA. And then we have our archaea. Then we have this sort of other branch which has coding DNA. Which has non-coding DNA. And from that, we've got the archaea. And we've got eukarya. Our eukaryotes, our nucleated cells. And within these eukaryotes, we still have our kingdoms. We still have plants, animals, fungi, and protists. So all that we've really done with this changing of information and different understanding from genetic analysis is we've just rewritten the rules a bit. So we've gone from 5 kingdoms to 3 domains. But within those domains, or within one of those domains in the eukaryotes, we still have, in our nucleated cells, remember bacteria don't have a nucleus, we still have 4 kingdoms. And that's all you really need to know. It's just this idea of why we changed things over, because it was so different. Because of these things, because archaea have these non-coding DNA, these promoters in between, and eubacteria don't. So we decide the archaea are actually closer to eukaryotes than they were to prokaryotes, or closer to eubacteria. So that is what the domains actually are. It's tough, it's difficult to remember, but now you've got a nice video that summarizes it all.